Hello. We were discussing that why DNA polymerase cannot synthesize in 3 in 3 dash to 5 dash direction and can only synthesize in 5 dash to 3 dash direction. Now what happens, let's say if I take a DNA strand and let's say its direction is, I'm taking a normal situation, 3 dash to 5 dash, A T G C A T A A G G, you can write anything, A T G T T G C. Now drawing its complementary strand, 5 dash A T A C G T, 5 dash to 3 dash. Now at 3 dash position we have OH. Now this is in DNA replication we are talking about. Nucleotide, this is nucleotides is there, 3 dash OH group is there, 5 dash phosphate is there, base is there, then sugar is there. Now let's say we have to add one upcoming. Now what happens, one nucleotide attaches with another nucleotide, then second with third nucleotide, then fourth nucleotide. So this way nucleotide comes, right? So now here we will talk about the upcoming nucleotide. Let's say this is the upcoming nucleotide, this has to be added. O, then P, O, then P double bond O, it's O negative, O, then P double bond O, O negative, and here it will be like this. So basically what happens here, we have a lone pair, this will attack on 5 dash phosphate, this is 5th position, this is the phosphate group present here, and here we have the DNA polymerase. So what will happen, a phosphodiester bond will be formed, then in pyrophosphate will be released, and an energy will be released so that a phosphodiester bond can be formed. This is the simple mechanism, or this is the normal procedure. Now let's say if I talk about a hypothetical condition, means 3 dash to 5 dash, which is not possible. Hypothetical case or situation. Earlier I have drawn from here, earlier it was 3 dash to 5 dash, I have written here. Now I am changing the directions. Earlier I wrote 3 dash to 5 dash, okay, and 5 dash to 3 dash. This was the correct scenario, but this one is actually the wrong scenario. But let's say if I'm taking a hypothetical situation, if it's from 3 dash to 5 dash, how will is it possible to take place? Let's see, A T G G C C, then A T T C G A T G C. Here it will come T A C C G G. Now at 5 dash position we have triphosphate. Now here this is the deoxyribose sugar. Ribose is 5-membered ring, hexose is 6-membered. So since it's deoxyribose, ribose is 5-membered, so this will be the structure. Let's say we have a base adenine. then this is the first position, this is the second position, H, third carbon, OH, third. We have to look see the third position. This will be the fourth carbon, and this will be the fifth carbon. How these positions are aligned, this is taught in biochemistry part. So at CH2, now here we'll form the PO negative, P double bond O, PO, then PO negative, double. I have just reversed the ring. I was drawing these double bond O upwards, now I'm drawing it downwards. Now what happens, let's say, because 3 dash group attacks, right? Here we have a lone pair. Now this will attack the 5 dash phosphate. Now what will happen? Let's say this, obviously this will, the bond will break between 3 dash OH and 5 dash phosphate, and pyrophosphate will be released and energy will be there. Now here we are left with monophosphate. Okay, now let's go to the next step. Only one phosphate is there, we are left with monophosphate. Now let's go to the next step. What happens? Coming to the concept of proofreading, again a hypothetical situation. Three dash, 5 dash, 3 dash. Okay, let's say then if I see the earlier one, here 3 dash to 5 dash we have taken here, 3 dash to 5 dash. This is the hypothetical, means incorrect ones, like we are assuming that thing. If it could happen, if it happens, what will be the result, like that. A T G C A T A G G C C, then A T, like so. Then it will be T A C G T G. Then again here A T G C A T A G G C C A T. Here it will come T A C G T G. So here we have 3 dash OH, and here we are having 5 dash. This will be, see, in normal case we have like triphosphate, but in the last step we have seen we are left with monophosphate. So let's say, then again here adenine is there, let's say adenine H, OH, CH2, then oxygen, then PO, O negative, then same structure. Now here we have lone pair. This will attack here. This bond will break down. So this is the normal condition. But in last case we have seen, let's say here we have adenine, structure same, then here CH2, and it will be so here I need to draw the rings such that it can come in, 3 dash will come like this. Here we have OH at third position, then this is the fourth carbon and this is the fifth carbon. Sorry, adenine will be here first, then second, then third, then fourth, and then it will be fifth here. CH2, OP, OP, and the structure goes like. So here we need to see this 3-OH. It has two lone pairs. Now it will attack this monophosphates. Here we don't have triphosphate because in the last step we have seen we were left with only one phosphate, right? So if it's monophosphate, is it possible to release? No pyrophosphates will be released, no energy will be released. If no energy will be released, that means no phosphodiester bond will be formed, and if no phosphodiester bond is formed, no nucleotide will be added. So that is why this DNA polymerase, this has it has proofreading activity. So it detects the like what is happening wrong. So this is the reason, like from we need we require triphosphate, right? But here we have, we are left with monophosphate. So since monophosphate condition is there, so it the synthesis is not possible from 3 dash to 5 dash direction. It's only possible from 5 dash to 3 dash direction. So basically the question is, why DNA polymerase cannot synthesize in 3 dash to 5 dash direction and can only synthesize in 5 dash to 3 dash direction. So the answer will be like because in 5 dash sorry, not in 5, because in 3 dash to 5 dash direction there is absence of triphosphate group, hence only we are left with, hence only monophosphate group is present. No phosphodiester bond is formed, so no new nucleotide will be added. Now why DNA, and here we'll write DNA polymerase has activity of, or you can say has proofreading activity. This word is must. Okay, so this was about it. Thank you.